When you step outside, especially if you live in a somewhat green area, there are certain things your brain just automatically categorizes as background scenery.
Oh, yeah, totally. Like a rock is a rock, a cloud is a cloud.
Exactly. And a tree is just a plant. I mean, a really big, quiet plant, but still just a plant. We walk past them every single day.
Right. We park our cars under their shade, build our houses out of them, rake their leaves.
Yeah, we treat them like familiar furniture of the natural world. I think we understand exactly what they are.
And that's, well, it's the ultimate example of familiarity, breeding a sort of profound collective ignorance. Because we see them so often, our brains use a cognitive shortcut.
We stop actually looking at them.
Exactly. We assume they operate on the exact same basic biological principles as, the grass we mow on the weekend or the potted fern wilting in the living room.
We just think of them as scaled up, like a tree is just a giant fern, right?
That is exactly the trap. But if you actually stop and look at the underlying biology, if you zoom in on the physics, the fluid dynamics, the molecular architecture,
Suddenly, that familiar background scenery becomes deeply, almost uncomfortably alien. And that is exactly what we're getting into today. Welcome to Audio Notebook.
glad to be here.
Today, our mission is to fundamentally shatter how you view forests. Because here is the mind-bending premise we're exploring. The towering trees you walk past are not really plants in the way you intuitively think of them.
No, they really aren't.
They are massive, hydraulically pressurized biocomposite structures built almost entirely out of shattered rock and atmospheric gas.
And we should clarify right at the top, that isn't poetic hyperbole. What's fascinating here is that the very condition of being a tree isn't actually a strict taxonomic category in evolutionary biology.
Wait, really? So you can't just point to one branch on the tree of life and say, these are the trees?
No, you can't. Being a tree is a biological strategy. It's a highly successful, repeatedly evolved strategy of vertical extension.
Okay, so it's a structural strategy, not a genetic family.
Right. And the detail that completely upends our perception is that a mature tree is almost entirely composed of dead matter. It's essentially a towering mountain of cellular corpses.
A mountain of corpses. I know that sounds incredibly dramatic, maybe a bit macabre, but as we're going to see today, it's the literal functional truth.
It really is. The only living part of that massive oak or that towering pine in your backyard is a microscopic, paper-thin cylinder of dividing cells.
It's this tiny, fragile fraction of life just completely trapped between a heavy outer armor of dead tissue and a massive internal skeleton of, well, more dead tissue.
So if we really want to understand how a tree works,
Yeah.
the physiological and molecular mechanics, we need a complete paradigm shift.
Right, we have to stop viewing them as mere plants soaking up sun. We have to start looking at them as highly engineered fluid dynamic miracles.
Exactly. These are organisms that manipulate the laws of physics to achieve a state of biological near immortality.
Okay, let's unpack this. Because to really understand why a tree is built this bizarre, morbid way, relying on dead tissue, we have to go back in time.
Way back.
Yeah, before trees or even land plants existed. We have to look at when the ancestors of all modern plants were, frankly, pretty squishy.
The story really begins with the aquatic ancestry of plant life. For over 1 billion years, the photosynthetic ancestors of modern plants lived exclusively in the water.
We're talking marine algae floating in the oceans and shallow seas.
Right. And from an engineering perspective, water is a profoundly forgiving medium. If you're an aquatic plant, your body can be incredibly delicate.
Because you don't need roots to search for water since you're literally swimming in it.
Exactly. You absorb water and dissolve nutrients directly through your entire surface. And crucially, you don't have to fight gravity.
It's like being an astronaut training in a swimming pool. You don't need strong leg bones to hold you up because the water's buoyancy is doing all the heavy lifting. You just float.
But then, roughly 470 million years ago, the Great Terrestrial Migration happened. Plants started creeping onto solid land. And the moment they left the water, the physics of survival completely changed.
Moving to land opened up an entirely new dimensional frontier, right? In the ocean, light only penetrates a few dozen meters down before it gets too dark.
Right. But on land, sunlight is intensely abundant. The issue is that the physical space to capture it is highly competitive.
And on solid ground, without that support of buoyancy, you face this relentless, invisible downward pull, gravity.
Gravity is the absolute enemy here. This inability to float transformed vertical height into the ultimate evolutionary weapon.
I love that framing. height as a weapon, because if you can just manage to grow a few inches taller than your neighbor, you get to monopolize the sunlight.
Yes, you become this giant solar panel, and you cast a shadow that literally starves the terrestrial competitors beneath you in the dark.
So it set off this massive evolutionary arms race for the sky. But early land plants had a major problem, didn't they? They were built primarily out of cellulose.
Yeah, cellulose was the critical structural bottleneck. At a molecular level, cellulose is a complex carbohydrate, a long-chain polysaccharide.
It's great for giving a plant cell its basic flexible shape, right?
It is. It has very decent tensile strength, meaning if you grab a strand of cellulose and pull on both ends, it resists snapping. However, it severely lacks compressive strength.
So it can't support massive vertical growth against the constant crushing weight of gravity.
Exactly. If you try to build a skyscraper out of wet spaghetti, it doesn't matter how much you stack. Eventually, it's just going to buckle and collapse under its own weight.
Cellulose alone wasn't going to win the arms race. They needed a new building material. And here is where it gets really interesting. The biological miracle that allowed plants to finally conquer gravity.
They evolved a molecule called lignin, and lignin is the absolute game changer for terrestrial life.
Biomechanically, what actually is lignin?
It's a highly recalcitrant three-dimensional heteropolymer. To use a human construction analogy, if those flexible cellulose microfibrils are the steel rebar inside a building's pillars.
Oh, then lignin is the concrete poured into the matrix around them.
Exactly. It fills all the microscopic interstitial gaps between the cellulose threads and the hemicellulose web.
And when that biological concrete hardens, it locks everything into place. It creates this rigid, incredibly tough composite material.
Right. Suddenly, the plant has unprecedented mechanical strength to push upward, and as a bonus, its internal plumbing becomes completely waterproof.
It's the equivalent of pouring solid concrete into a world that was previously made of jelly. But a plant doesn't just go to the hardware store for concrete. How does a living cell manufacture something this complex?
The biochemistry behind lignin synthesis is staggering. It's synthesized through the enzymatic dehydrogenation of three primary phenolic building blocks.
Okay, those base units are known as monolignules, right?
Yes.
Yeah.
Specifically, they're p-coumaryl alcohol, coniferyl alcohol, and synapyl alcohol.
Those are some heavy chemical names. Picomeryl, coniferyl, and synapyl alcohols. I'm just picturing them as three different, highly specialized types of microscopic Lego bricks.
That's a good way to look at it.
But how does the plant press those bricks in the first place?
It relies on a metabolic assembly line known as the phenylpropanoid pathway. It's this elegant, step-by-step molecular factory. The raw material entering the factory is a very common, simple amino acid called phenylalanine.
Okay, so phenylalanine goes in. What's the first step on the line?
The very first step is a process called deamination. The plant uses a specific enzyme to slice the nitrogen-containing alanine group right off the phenylalanine molecule.
So it's basically stripping the amino acid down to its chassis.
Exactly. That initial stripping transforms it into cinnamic acid. From there, the molecule moves down the line through a cascade of successive enzymatic reductions and hydroxylations.
So the cellular machinery carefully tweaks the molecule, adding an oxygen here, removing a hydrogen there, step by step.
Right, until it finally yields those three complex monolignyl alcohols. Then, those alcohols are exported to the cell wall, where they polymerize. They lock together in a chaotic three-dimensional web to form the solid lignin concrete.
It's an incredibly sophisticated manufacturing process. But here is the wild evolutionary theft that completely blew my mind.
Oh, the origins of the pathway, yeah.
Right. You'd logically think that this complex genetic architecture to build lignin evolved specifically because plants needed to stand up on land, right? Like they felt gravity, they needed a solution, so they invented concrete. Necessity is the mother of invention.
That is the most intuitive assumption. But the genomic record tells a completely different, almost paradoxical story. The genetic architecture required for lignin biosynthesis actually significantly predates the migration of plants onto land.
This is the part I find so wild. The enzymes required to synthesize picomoral alcohol, that very first crucial Lego brick of lignin, they didn't first appear in towering trees.
No, they didn't. They already existed in various photosynthetic eukaryotes that lived exclusively in the water. We're talking about marine diatoms, ancient red algae, and green algae.
The obvious question then is, if these ancient aquatic organisms were floating in the ocean, completely supported by water, standing tall wasn't their goal, gravity wasn't an issue, so why did they possess the genetic blueprints for biological concrete?
It turns out, in the ocean, these specific phenolic compounds served an entirely different purpose. They likely functioned as powerful ultraviolet protectants.
So they were essentially biological sunscreen.
Sunscreen and also potent antimicrobial defenses against marine pathogens. They absorbed harmful UV radiation that could mutate DNA, and they poisoned attacking microbes. They had absolutely nothing to do with structural load-bearing support.
It's an incredible example of evolutionary repurposing, taking a chemical sunscreen from an ocean algae and turning it into the concrete foundation of a redwood. But wait, it goes even deeper than the algae. Where did the algae get those specific enzyme blueprints in the first place?
Deep phylogenetic analyses point to a mechanism that often surprises people: lateral gene transfer. It appears these genetic blueprints were originally acquired from soil-dwelling bacteria.
So visualize the timeline. A genetic sequence that originated in ancient bacteria was passed laterally to aquatic algae, where it was utilized for UV protection.
Yep. Then, hundreds of millions of years later, as the descendants of those algae crept onto land, they co-opted that exact same genetic machinery. They upregulated it, tweaked the final polymerization step, and used it to build the very first wood.
Talk about standing on the shoulders of microbes. So, armed with this stolen bacterial sunscreen turned biological concrete, early land plants start pushing upward. They start testing the limits of gravity.
And by the late Devonian period, which is roughly 385 million years ago, plants have gone all in on this lignin strategy. The late Devonian marks a fundamental transition in Earth's history.
It's the era that sees the emergence of the first true wood, right? Which botanists classify as secondary xylem.
Exactly. The organism that really pioneered and perfected this strategy was a massive progymnosperm called Archaeopteris.
If you were to travel back in time and look at Archaeopteris, you would immediately recognize it as a tree, wouldn't you?
Oh, absolutely. It deployed advanced horizontal lateral branching to capture light, and it developed deep, complex woody root systems. It fundamentally transformed the terrestrial landscape from a flat, two-dimensional crust into a towering three-dimensional biosphere.
And this brings up a massive geological misconception that we really need to clear up today. Because the old textbook story, the one I definitely learned in school, was that the sudden appearance of these massive, towering Devonian forests acted as a giant vacuum.
Right, the carbon drawdown narrative.
Yeah. The narrative was that these massive new trees sucked all the carbon dioxide out of the atmosphere to build their huge wooden trunks, and this massive carbon drawdown caused a global anti-greenhouse effect. It plunged the Earth into an ice age with massive polar glaciation.
It's a very neat, compelling narrative, and it highlights the power of biology over climate. But recent, highly sophisticated biogeochemical analyses of fossilized plant material suggest the reality is much more nuanced.
Okay, so what actually happened?
We now have geochemical data indicating that the initial massive crash in atmospheric carbon dioxide actually occurred millions of years before the large woody forests of Archaeopteris evolved.
Wait, so the giant trees didn't cause the initial climate crash. Who did?
It was driven primarily by primitive, early vascular plants that were much smaller, more akin to low-lying woody shrubs. They were the ones that initially covered the land masses and began the aggressive chemical weathering of silicate rocks.
Which is the primary mechanism for drawing down long-term atmospheric CO2.
Exactly. By the time the giant canopy-forming trees evolved, the atmospheric carbon had already substantially crashed.
But the giant trees arrived at a party that was already cooling down. So what did the evolution of the massive tree actually do to the planet, if not freeze it?
The profound impact of large trees was ecological and geological, rather than purely climatic. As I mentioned, they established deep, three-dimensional ecosystems, providing vertical habitats for the subsequent explosion of terrestrial animal life.
And their root systems played a big part too, right?
Oh, absolutely. Their immense, powerful, lignin-reinforced root systems fundamentally altered the Earth's terrestrial weathering cycles. They physically fractured and broke apart the solid bedrock deep underground, accelerating soil formation.
And crucially, their massive, rot-resistant lignin bodies introduced a massive new carbon sink, storing deep organic carbon directly into the lithosphere for the first time. They didn't start the cooling, but they changed the physical and chemical architecture of the Earth's crust forever. Okay, so we've established the why and the when. Plants needed to beat gravity to get sunlight, so they repurposed stolen bacterial sunscreen genes to make biological concrete, fundamentally reshaping the Earth's crust in the process. It's a phenomenal origin story.
It really is.
But now we have to look at the how. How does a tree actually coordinate the building of a massive lignin-reinforced skyscraper? If I look at a giant redwood, where is the brain? Where is the command center directing the construction?
The command center of a tree is an anatomical structure known as the vascular cambium. And what is most remarkable about the cambium is how physically insignificant it appears to the naked eye.
I mean, if you were to peel back the bark of a mature tree, what would you see?
The cambium is just a razor-thin, cylindrical meristematic zone. We are talking about a layer of stem cells that, in many cases, is only a single cell wide, or perhaps just a few cells across.
Yet this microscopic cylinder stretches continuously from the very tips of the deepest roots all the way up into the highest, finest branches in the canopy. Just visualize that for a second for you
That's
listening.
what I think about.
A cylinder of active life, practically invisible, just a few cells thick, but stretching for hundreds of feet, actively building a redwood from the inside out. It's almost impossible to comprehend. You called it a bifacial stem cell niche. What does bifacial actually mean in terms of how it builds?
Bifacial refers to the fact that this single layer of cells operates in two opposing directions. The vascular cambium is the biological engine responsible for all radial growth, meaning it dictates how the tree increases its diameter and gets wider over time.
Okay, so as these cambial stem cells divide, the newly created daughter cells have two potential developmental fates.
Correct. If a daughter cell is produced and pushed toward the interior of the tree, the proximal domain, it differentiates into secondary xylem. That is the tissue we commonly refer to as wood.
Okay, so pushing inside makes wood.
Right. Alternatively, if the stem cell divides and pushes the daughter cell toward the exterior of the distal domain, that cell will differentiate into secondary phloem. That's the conductive tissue that eventually gets crushed outward to become part of the inner bark.
So the cambium builds wood on the inside and bark on the outside, constantly expanding the perimeter of the tree. It's like a microscopic cylindrical 3D printer.
That's a perfect analogy.
It's constantly printing a rigid skeleton on its inner surface and printing a flexible suit of armor on its outer surface, all while slowly expanding outward as the skeleton grows. But here is the critical question.
How does it know what to do?
Yes. How does a single microscopic stem cell know which tissue to become? If it gets pushed a fraction of a millimeter to the left, it becomes a water pipe in the wood. If it gets pushed a fraction of a millimeter to the right, it becomes a sugar pipe in the bark. How is that immense architectural decision made without a brain?
That spatial decision-making process is strictly, rigorously governed by overlapping, highly complex gradients of phytohormones and highly specific gene regulatory networks. It's an intricate chemical conversation happening across a distance of micrometers.
Okay, so what are the primary signaling molecules driving this?
The primary morphogens, the signaling molecules that dictate form and fate coordinating this entire spatial organization, are a class of hormones called auxins, specifically indole-3 acetic acid, and another class called cytokinins.
Let's break those down because hormones can be abstract. What is auxin actually doing in this tiny razor-thin cylinder?
High concentrations of auxin are absolutely essential for maintaining the pluripotency of the cambial stem cells.
Pluripotency meaning the cell hasn't committed to a final fate yet.
Exactly. It remains young, flexible, and capable of dividing into anything. The cambium maintains a very tight, localized peak of auxin. The specific concentration gradient of auxin, as it diffuses radially away from that cambial center, dictates the ultimate fate of the differentiating daughter cells.
But auxin is a blunt instrument. It doesn't work alone.
No, cytokinin signaling is equally critical for spatial precision.
So what does the cytokinin do?
Cytokinin acts as a boundary enforcer. Modulated by a specific internal inhibitor known as AHP6, cytokinin restricts the proliferative, dividing stem cell activity strictly to that central cambial domain.
So it ensures that the explosive growth potential of these stem cells remains tightly organized in a neat cylinder.
Yes, preventing it from spiraling out into a chaotic, disorganized mass of tumorous tissue.
So you have auxin keeping the stem cells young and flexible in the middle, and cytokinin acting like the guardrails, keeping them strictly in their lane so they form a perfect ring. It's a microscopic balancing act.
It really is.
And my understanding is that our ability to actually see this happening has completely exploded in the last few years, right?
Absolutely. Recent massive advancements in a technology called single-cell RNA sequencing have allowed molecular biologists to observe this differentiation process with unprecedented, almost terrifying resolution.
We can now essentially read the active genetic diary of a single cell as it makes these fate decisions.
We can. And what we see is the extreme rapidity and precision of the process. As a daughter cell is pushed just a microscopic distance, literally 1 or 2 cell widths away from that central stem cell niche, its developmental trajectory transitions almost instantaneously.
Okay, let's trace the life of one of these cells to see how fast it happens. Let's look at a cell that gets pushed to the outside the distal domain. It's moving out toward the surface. It's destined to become phloem, the inner bark. What physically flips the genetic switch inside that cell?
The lineage bifurcation into phloem tissue is driven by a highly specific family of transcription factors known as pair.
And pair stands for, let me check my notes here, phloem, early DNA binding with one finger. Biologists really have a knack for poetic names.
They do try.
So what is this pair factor actually doing?
It acts as the trigger for bark creation.
Yeah.
However, within the central stem cell niche and in the immediate transit amplifying cells right next to it, pair activity is strictly locked down.
How is it locked down?
It is continuously actively suppressed by a concentration gradient of another set of transcription factors called plethora, which emanate from the very core of the cambium.
So think of plethora as a localized radio jamming signal. As long as the cell is near the center, the jammer blocks the pair signal.
That is a perfect way to visualize it. But as ongoing cell division in the cambium continually pushes our daughter cell further and further outward, it physically moves away from the source of that plethora gradient.
The concentration of the jammer drops, the jamming signal dissipates.
Exactly.
And the moment that plethora jamming signal fades away, boom, the pair transcription factors are liberated. They wake up from their suppression.
Exactly. And once liberated, the pair factors don't waste time. They immediately proceed to bind to the DNA and activate a cascade of downstream genetic targets.
And the most critical of these is the altered phloem development gene.
Race. Once this specific gene is activated, the point of no return is crossed. The cell is genetically locked in. Its fate is sealed. It enters a dedicated phloem differentiation trajectory.
Which, as we're going to discuss in detail later, involves an incredibly dramatic, almost horrific physical transformation. So that is the genetic lock-in for the phloem on the outside. But what about the inside? The cells that get pushed inward into the proximal domain, the ones destined to become the xylem, the solid wood.
Xylem differentiation is driven by an entirely distinct, highly complex suite of transcription factors. But what is crucial to grasp here is not just the specific names of the genes, but how the two systems interact.
What do you mean?
These two genetic networks, the xylem-promoting genes on the inside and the phloem-promoting genes on the outside, actively and aggressively suppress each other.
Right, they fight each other.
Yes, it creates a robust seesaw mechanism of mutual genetic inhibition. If the xylem genes start turning on, they immediately send signals to ensure the phloem genes stay permanently off, and vice versa.
That mutual hostility ensures absolute spatial segregation. It guarantees that a given cell differentiates definitively into either a water-conducting wood cell or a sugar-conducting bark cell, but never a confused, non-functional mixture of both. Exactly.
That is brilliant. This razor-thin layer of cells is constantly executing this incredibly complex hormonal and genetic calculus millions of times a day across the entire surface area of the tree. But here's the thing.
Right, it's not in a vacuum.
Exactly. It isn't doing this math in a sterile laboratory. It's doing it outside, exposed to the chaotic weather. And that brings us to a phenomenon that every single person listening has definitely seen with their own eyes. Tree rings.
A classic feature.
What does all this microscopic, unseen hormone signaling have to do with the rings we count on a sawn stump?
They are the exact same phenomenon, just viewed at different scales. That delicate microscopic hormonal balance within the vascular cambium is profoundly, acutely sensitive to macroscopic environmental signals.
So the cambium is essentially a biological sensor array. Precisely. It reacts continuously to seasonal fluctuations in ambient temperature, photoperiod, which is the length of daylight, and soil water availability. These external factors directly dictate not just how fast the cambium divides, but the actual physical geometry and thickness of the cells it manufactures.
Let's walk through a year in the life of the cambium to see how this works. Let's say spring arrives. The winter snow melts, the soil is saturated with water, the days are getting longer and warmer, the tree is waking up. What does the cambium do?
During the highly favorable, resource-rich conditions of spring, the tree's primary physiological priority is to move as much water as physically possible from the roots up to the newly flushing canopy of leaves to support rapid growth.
Makes sense. It's thirsty.
To accomplish this, the cambium shifts its manufacturing parameters. It responds by producing large, wide-diameter xylem cells with relatively thin walls.
It builds massive water pipes.
Precisely. These specific cells are optimized purely for maximum internal volume and rapid low-friction fluid transport. Because these cells are mostly empty space with thin walls, when you look at a cross-section of the wood, this layer appears as a wider, highly porous, visibly lighter colored band.
And botanists refer to this specific growth as early wood. Okay, so spring early wood is all about big, wide pipes for maximum flow. But spring doesn't last forever.
No, it doesn't.
Summer hits, the days get intensely hot, rain becomes scarce, the soil begins to dry out, the tree is starting to experience environmental stress. How does the factory respond?
When water becomes scarce, the physics of pulling water up to the canopy change dramatically. The hydraulic tension inside the tree, the suction increases exponentially.
We're going to cover the mechanics of this section later, but for now, those wide, thin-walled spring cells are suddenly highly vulnerable, right?
Yes. Under intense suction, they are at risk of physically imploding or collapsing under the strain.
Like sucking too hard on a cheap plastic straw.
That's a perfect analogy. So, the environmental signals of heat and drought cause a rapid hormonal shift within the cambium. It recognizes the danger and shifts its production line.
It stops making early wood.
Right. And it begins to produce what is known as late wood. These are completely different looking cells. They are much narrower in diameter, and they feature heavily lignified, extraordinarily thick secondary walls.
So they're not designed for maximum water flow. They're designed specifically for extreme mechanical strength, to resist the immense physical tension caused by drought.
Exactly. And because these late wood cells are so narrow and their walls are packed so thickly with dense lignin, when you look at them, they appear as a much darker, much denser, thinner line to the naked eye.
So when you look at a tree stump and you see a light, wide band followed by a dark, thin line, you are quite literally looking at the physical manifestation of spring turning into late summer.
You are.
You're seeing the tree's hormonal panic response to a drying environment permanently set in biological concrete.
This environmentally modulated oscillation in cellular geometry generates the visible, alternating annual growth rings. It's a profound realization. The tree is passively embedding a highly accurate year-by-year, high-resolution climatic history directly into its own structural architecture.
It's an environmental hard drive. But let's pause and look at what is actually happening on a cellular level to these inward-facing cells. The ones destined to become the wood, the early wood and late wood we just talked about.
The trade tree elements.
Right, because we've set them up as the structural water-conducting backbone of the tree. But what a single cell actually goes through to achieve that status is, frankly, horrifying.
It is a stark, almost brutal reality of plant biology. The inward-facing specialist cells produced by the cambium embark on what can only be described structurally as a conveyor belt of death.
A conveyor belt of death.
Yes. To function as an efficient hydraulic pipeline, a secondary xylem cell cannot possibly remain alive. Think about the internal architecture of a living cell. It is packed full of viscous cytoplasm, massive organelles like vacuoles and chloroplasts, a bulky nucleus, and a dense cytoskeleton.
Right. You can't efficiently pump water through a pipe that is already stuffed full of thick jelly and microscopic machinery. The pipe has to be entirely empty to work, so the cell has to die.
Yes.
But it doesn't just quietly starve to death in the dark, does it?
Not at all. The process, scientifically termed phylogenesis, is a highly orchestrated, terrifyingly precise form of vacuolar programmed cell death. It fundamentally challenges traditional, simplistic notions of cellular biology because it demonstrates how a living cell meticulously, intentionally arranges its own post-mortem functionality.
The execution of xylogenic programmed cell death is not passive decay. It's an actively regulated, highly energetic, altruistic suicide. Altruistic suicide. The cell is willingly, actively destroying itself for the functional good of the whole tree.
Yes, that's exactly what it's doing.
Let's get into the biochemistry of this cellular execution. How does a cell actually manage to kill itself with such precision that it leaves a perfect pipe behind?
It is governed by a specific set of molecular master switches. The most notable of these are a family of transcription factors known as the vascular-related NAC domain, specifically the genes VND6 and VND7.
When the differentiating cell reads those specific lines of genetic code, it knows its time is up.
Right. It enters its terminal phase, and ironically, it goes into metabolic overdrive.
It's like a frantic final act of extreme productivity. What is it building in its final moments?
Two things simultaneously. First, it rapidly synthesizes robust, incredibly strong secondary cell walls, packing them intensely with the lignin we discussed earlier.
It's actively building the reinforced walls of its own tomb to ensure they won't collapse under the massive hydraulic pressures after it's gone.
Precisely. And simultaneously, the cell begins accumulating massive quantities of highly destructive hydrolytic enzymes.
Destructive enzymes, we're talking about biological dismantling tools.
Yes. It manufactures endonucleases, designed specifically to shred its own DNA, ribonucleases to destroy its RNA transcripts, and metacaspases designed to systematically dismantle its structural proteins.
And where does it put all this dangerous stuff?
It carefully stockpiles all of these deadly enzymes inside its large central vacuole, which acts like a secure, pressurized biological vault in the center of the cell.
So just to visualize this, it builds an indestructible outer concrete wall, and then it builds a massive chemical bomb inside itself. A bomb made of highly corrosive digestive enzymes. And then, what pulls the trigger?
Once the structural lignification of those outer walls is absolutely structurally complete and not a moment before an internal molecular signal is triggered.
What's the signal?
Biologists are actually still working to understand the exact biochemical nature of this final trigger, but the resulting event is dramatic and irreversible. It causes the sudden catastrophic rupture of the tonoplast. The tonoplast is the vital membrane enclosing that central vacuole wall.
So the bomb goes off.
Exactly. The simultaneous arrest of all living cytoplasmic streaming gives way to rapid, chaotic autolysis. The highly acidic, enzyme-rich contents of the vacuole forcefully flood out into the cytoplasm.
And they systematically and aggressively digest the nucleus, the mitochondria, the plastids, the entire cytosol.
It completely clears the protoplast, dissolving all the biological machinery that built the cell in the first place.
It literally dissolves itself from the inside out. But there's a really important biological distinction you made there. The whole process isn't just one thing called death.
Right. Plant biologists make a crucial, highly specific distinction regarding this terminal phase. There is the programmed cell death, which refers exclusively to the highly regulated sequence of gene activation, massive enzyme synthesis, and structural reinforcement leading up to the arrest of life via that tonoplast rupture.
And then there is the autolysis.
Yes. Autolysis is a purely biochemical cleanup process. It's the physical, enzymatic digestion and clearing of the cellular debris.
So the programmed cell death is the careful sentencing and the final execution, and the autolysis is the cleanup crew coming in to mop the floors. And what remains after this intense, violent autolysis is complete.
Just an empty, heavily fortified, microscopic tube.
And the cambium just keeps making them, layer upon layer, day after day, year after year. These cellular corpses are stacked vertically and radially, generating this massive biological graveyard of literally trillions of plant bones. That is what the structural core of a massive tree actually is.
It is a profound, almost philosophical realization when you truly grasp the scale of it. But there is a massive twist to the story regarding the very center of the tree, the deep wood.
Oh, right, the heartwood.
Because for a very long time, the old textbook model of a tree's interior assumed a very passive, inevitable process of decay. The model was, you have the outer sapwood, which is comprised of these newly dead, empty tubes actively conducting water.
And as the tree ages, those inner rings of sapwood slowly get clogged up with air or resins. They stop conducting water, and they just passively chemically decay over decades into what we call heartwood, the dark, incredibly dense central pillar you see in an old stump.
Right.
I remember learning exactly that. The heartwood is just old, retired, slowly rotting sapwood. It's the dead center of the dead wood. But that entire model is completely wrong, isn't it?
It is completely fundamentally inaccurate. Recent molecular and transcriptomic analyses of deep wood cores have completely overturned this passive assumption.
So what's really happening?
The formation of heartwood is an incredibly active, metabolically expensive, and highly regulated physiological process. It is the absolute opposite of passive senescence or rot.
Wait, hold on. If the sapwood is entirely made up of those xylem cells we just watched commit highly orchestrated altruistic suicide, how can heartwood formation possibly be an active process? Dead, empty tubes cannot have a metabolism.
You've hit on the core paradox, and the answer lies in a different cell type. You are correct that the axial tracheary elements, the vertical water pipes, die very quickly, but they are not the only cells embedded in the wood matrix.
What else is in there?
There is another vital network of cells called ray parenchyma. These are horizontal, radial cellular conduits. They run from the inner living bark, piercing straight through the cambium and extending radially deep into the center of the wood.
And crucially, these ray parenchyma cells do not commit suicide. They remain alive, metabolizing and functioning deep inside the dead sapwood.
Exactly.
Ah, so there are living, breathing survivors scattered deep inside the graveyard. So what do these living ray parenchyma cells do when it's time to transform that area into heartwood?
As these living parenchyma cells age and physically reach the sapwood-heartwood transition zone, buried deep inside the dark trunk, they experience a sudden, massive surge in metabolic activity.
This awakening is initiated by complex internal signaling, right?
Yes, which research heavily implicates to be driven by the accumulation of the gaseous hormone ethylene. When they receive this prolonged ethylene signal, these aging cells wake up and rapidly upregulate the genetic synthesis of extremely potent secondary metabolites.
They start actively manufacturing chemical weapons deep in the dark. What kind of weapons are we talking about?
They synthesize a vast, complex arsenal of defensive compounds. We see high concentrations of flavonoids, complex heavy tannins, and highly specific toxic lignans, including powerful antifungal molecules like saponin and Taiwanin A.
Saponin and Taiwanin A. These are incredibly complex, highly toxic compounds. Why are these few living cells suddenly pumping them out?
Their ultimate objective is to chemically fortify the dead core of the tree against fungal and bacterial decay. The tree needs a solid pillar to stand on. However, there is a fatal catch.
What's the catch?
The massive accumulation of these highly toxic antimicrobial compounds inside the living ray parenchyma cells themselves induces severe, inescapable oxidative stress.
The environment simply becomes too toxic for the cells to survive their own biological creations.
Yes. This internal toxicity inevitably triggers a delayed, final phase of programmed cell death, specifically ending the life of the ray parenchyma.
So they essentially poison themselves to death to protect the inner column of the tree. That is incredible. The dense, durable, beautiful, dark center of an old oak or a redwood, the heartwood that resists rot for centuries after the tree is cut down, is not the result of passive decay.
Not at all.
It's the result of a terminal, suicidal chemical defense deployment by the absolute last living cells in the core of the tree.
Precisely. It is a calculated evolutionary sacrifice. They die to create an impregnable, rot-proof structural pillar that will support the entire living perimeter of the organism for centuries, or in some cases, millennia.
Okay, if we connect this all back to the bigger picture, the tree has now successfully built this massive, heavily fortified pipeline comprised of trillions of dead, empty corpses. The outer walls are reinforced with biological concrete. The center is pumped full of toxic tannins and lignans to prevent rot. It's an impenetrable, towering fortress.
And a highly efficient one.
But its fundamental daily job is to move water from the dirt to the sky. And here is where I always struggle with the physics of a tree. How does an organism move water up to 113 meters into the sky, which is the height of the tallest coastal redwoods, without a single mechanical pump, without a beating heart?
The sheer physical scale of this fluid transport is staggering, and it defies our intuition. To contextualize it on a cellular scale, moving water 113 meters vertically through microscopic tubes is akin to a human attempting to operate functionally while their biological resources, their blood, their oxygen, the nutrients, are scattered across different continents.
It seems mechanically, thermodynamically impossible. If I try to suck water up a plastic straw that is taller than a couple of stories, the physics just break down. You literally cannot pull a vacuum strong enough with a mechanical pump. The water just stops rising. So how do trees bypass this fundamental limit of suction?
The mechanism utilized by plants to achieve this seemingly impossible vertical lift is known in biophysics as the cohesion tension theory. It was originally proposed by two brilliant Irish scientists, Henry Dixon and John Jolly, way back in 1894.
And despite immense initial skepticism from the scientific community, it's been rigorously validated by over a century of extensive biophysical research, right?
Yes, it has. This elegant system relies entirely on two fundamental phenomena: the immense thermodynamic energy provided by the sun and the inherent, almost magical chemical properties of water molecules themselves.
Let's start with the water molecule. What makes water so physically special that it can perform this trick?
It comes down to polarity. Water molecules are highly polar. They have a slight positive charge on one end and a negative charge on the other. Because of this, they were incredibly cohesive. They constantly form strong, dynamic hydrogen bonds with each other. They magnetically cling tightly to one another.
And they're highly adhesive too, right?
Yes, they adhere tenaciously to the hydrophilic, heavily lignified internal walls of those dead xylem conduits we discussed. Because this extreme internal cohesion and external adhesion, Water inside a tiny microscopic capillary tube doesn't act like a collection of individual free-flowing molecules.
It physically acts like a continuous, unbreakable, solid liquid rope.
Exactly.
A liquid rope. I absolutely love that visualization for you listening. So you have this unbroken microscopic string of chemically bonded water molecules stretching all the way from the tiny root hairs deep in the soil, up through the labyrinth of dead xylem tubes, all the way to the microscopic veins in the leaves high in the canopy.
Yes.
But a rope still needs to be pulled. Where is the immense pulling force coming from if the tree has no pump?
The pulling force, the engine of the entire system, comes directly from the sun. As solar radiation heats the canopy of the tree, the water inside the leaves naturally evaporates. It transitions from a liquid to a gas and exits the leaf into the atmosphere through billions of microscopic adjustable pores called stomata.
And this continuous solar-driven evaporation process is known as transpiration.
Right. Astonishingly, up to 95% of all the water a tree expends energy to absorb from the soil is simply evaporated instantly out into the atmosphere.
95%. That seems so incredibly wasteful for an organism to just throw away almost all of its water, but you're saying that waste is actually the engine itself.
Yes, because that phase change evaporation creates a severe negative pressure or tension at the very top of the water column within the leaf. As one single water molecule evaporates and leaves the stomata, its hydrogen bond physically pulls on the water molecule immediately below it.
And because the entire column of water is chemically linked together like that liquid rope, this microscopic atomic pull at the top is transmitted instantaneously, molecule by molecule, all the way down the massive trunk to the deepest roots.
The thermodynamic energy of the sun is quite literally grabbing the top of the liquid rope and pulling it continuously upward into the sky.
But the physical forces involved here must be astronomical. To lift a column of liquid water over 100 meters straight up against the continuous downward pull of gravity, plus overcome the immense friction of moving through those tiny, rough, microscopic tubes. How much actual pressure are we talking about here?
The physics are indeed staggering. To achieve this continuous lift, the transpiration happening in the leaves must generate suction forces equivalent to dozens of atmospheres of negative pressure. In tall canopy trees, the tension within the xylem routinely exceeds 1.9 to 3.0 megapascals.
Okay, let's put 3.0 megapascals of negative pressure in perspective for a second. The positive pressure inside your car tire is maybe 0.2 megapascals. This is 15 times that physical force, but pulling in the exact opposite direction.
It's immense.
It's like the crushing pressure you feel at the bottom of the ocean, but completely reversed into an agonizing pulling vacuum. What happens to liquid water when you put it under that much sheer tension?
That is the crux of the entire evolutionary engineering problem. Under such extreme negative pressure, water is stretched so thinly that it exists in a highly metastable state. According to standard fluid dynamics, if a human engineer built a steel machine that attempted to generate this much suction on a column of liquid water, the water would instantly fail.
It would boil.
Yes, it would explosively vaporize from a liquid into a gas at ambient temperature, purely due to the vacuum. This physical phenomenon is known as cavitation.
You literally pull so hard on the water molecule that you rip the liquid apart into gas.
Exactly. And when cavitation occurs inside a tree's microscopic xylem conduit, a sudden vapor bubble, an embolism forms within the tube. This expanding vapor bubble instantly snaps the hydrogen-bonded liquid rope.
And once that delicate rope is snapped, that specific microscopic tube permanently loses its ability to transmit the transpirational pull. The hydraulic line is dead.
It is dead.
If one tiny bubble can snap the rope, and the tree relies on thousands of these microscopic pipes, all held under insane physics-defying tension, how does the whole system not just catastrophically fail on a hot summer afternoon? The sun beats down, the suction peaks, a few bubbles form, the vacuum drops, and the whole 300-foot tree dies of thirst in an hour.
That is the constant existential risk every tree lives with every single day. But trees mitigate this catastrophic risk through incredibly precise microscopic anatomical adaptations. First, the xylem conduits themselves are evolved to be extremely narrow.
Because narrower tubes drastically increase capillary stability, making the cohesive water column structurally harder to snap.
Yes. But the true genius of the system lies in how these thousands of conduits are interconnected. They are not isolated. They are linked by microscopic structures known as pit membranes.
Pit membranes. What are those, and how do they stop the bubbles?
Think of pit membranes as microscopic, highly sophisticated safety valves located in the walls between adjacent water pipes. They are reinforced, highly porous biological membranes.
Their primary function is to allow liquid water to pass freely and easily laterally between adjacent vessels so the water can constantly detour around temporary blockages, right?
Exactly. However, the microscopic pores within these membranes are physically, meticulously sized by evolution to be just slightly too small to allow an expanding gas bubble to pass through.
Wow. So if the tension gets too high, an embolism forms and a pipe suddenly fills with gas. The bubble tries to rapidly expand into the next pipe to destroy that one too, but it gets physically trapped and contained by the microscopic mesh of the pit membrane. The safety valve slams shut.
Precisely. This brilliant isolation confines the catastrophic embolism to a single, localized cellular chamber. The liquid rope in that one specific tube is irreparably broken, but the integrity of the vast surrounding hydraulic network is perfectly preserved.
The transpirational pull simply draws the water laterally, detouring it around the dead, gas-filled pipe through the pit membranes of the adjacent, still-functioning conduits.
Exactly.
It's exactly like a submarine automatically sealing off a flooded compartment with bulkhead doors to save the rest of the ship. But if this happens often enough, and it must happen all the time in summer over years and years, wouldn't the tree eventually just accumulate too many embolisms? Wouldn't all the pipes eventually fill with gas until there's no path left?
For a very long time in plant physiology, we assumed exactly that. We believed that xylem transport was a purely passive, thermodynamically downhill process, and that once a vessel cavitated, it was permanently, irrevocably lost to the tree.
But recent groundbreaking technological findings have fundamentally challenged this notion, haven't they?
Yes, we now have concrete evidence indicating that trees can actively repair embolisms. They can actually refill cavitated gas-filled vessels with liquid water, even while the bulk of the xylem surrounding them remains under extreme pulling negative tension.
Okay, stop. That sounds physically impossible. How can a plant possibly push liquid water back into a gas-filled tube when all the functioning tubes immediately around it are pulling with 3 megapascals of vacuum? The vacuum in the adjacent tube should just instantly suck any new water right back out.
It is an incredible, almost unbelievable feat of active uphill biological secretion. And the mechanism relies heavily on those living cells we mentioned earlier, the parenchyma cells that live scattered adjacent to the dead xylem conduits.
When an embolism is detected, these living parenchyma cells jump into action.
Right. They utilize highly specialized proteins embedded in their cell membranes called cation chloride cotransporters. Using substantial amounts of cellular energy ATP, they actively pump mineral ions, essentially salts, out of their own cytoplasm and directly into the dead, embolized, gas-filled xylem conduit.
So they are actively pumping salt into an empty dead pipe. What does that achieve?
It creates a severe, highly localized osmotic gradient. By making the inside of the embolized tube highly concentrated with mineral ions, it creates an immense chemical draw for water.
And this powerful osmotic pull actually becomes stronger than the physical negative tension of the surrounding vascular system.
Exactly. Water is literally forcefully dragged back into the gas-filled chamber from surrounding tissues through specialized water channels called aquaporins. The rising tide of water inside the tube forcefully compresses and dissolves the gas bubble back into solution, ultimately restoring the continuous liquid rope and reestablishing hydraulic continuity.
That is absolutely mind-blowing. The tree isn't just a passive set of pipes, it is constantly monitoring its microscopic plumbing, dynamically sealing off broken pipes, pumping them full of salt, and forcefully dissolving gas bubbles against massive thermodynamic gradients. It is an active, hyperdynamic hydraulic machine.
It truly is.
But even with all of this incredible, intricate engineering, there is still a hard limit, right? Trees are tall, but they don't grow to the moon.
No, they do not. Despite these phenomenal anatomical safeguards and active osmotic repair mechanisms, the sheer physics of hydraulics represent the ultimate biological ceiling on tree growth. This physical reality is formalized in what botanists call the hydraulic limitation hypothesis.
Okay, break down the hydraulic limitation hypothesis for us. Why does a redwood stop at exactly 113 meters?
It posits that as a tree grows progressively taller year after year, two compounding physical challenges arise. First, the physical path resistance increases. Pushing liquid water through 100 meters of microscopic, friction-heavy tubing is exponentially harder than pushing it through 10 meters.
And second, the sheer gravity-induced weight of the continuous water column increases massively.
Yes. To overcome both the friction and the weight, the tension at the pot, the suction force generated by the leaves must become increasingly severe.
The taller the tree gets, the harder the leaves have to pull just to get a drop of water.
Correct. And eventually, a critical, fatal threshold is reached. The suction required becomes so immense that the risk of runaway, systemic cavitation, the risk of the entire liquid rope system flashing into vapor under the immense strain becomes too high.
To prevent this catastrophic failure, the tree is forced to defensively close its stomata earlier and earlier in the day when the sun gets hot.
Right. It has to stop the evaporation to drop the suction and save the water column. But there is a huge, unavoidable trade-off there.
A massive growth-limiting trade-off, because the stomata on the leaves are not just the exit valves for water vapor, they are the primary intake valves for carbon dioxide from the atmosphere.
Exactly. When the stomata clamp shut to protect the hydraulics, the vital influx of carbon dioxide is immediately choked off. Without carbon dioxide, the chemical engine of photosynthesis halts. The tree can no longer manufacture sugars.
Therefore, this protective hydraulic closure ultimately caps the total energy production of the tree, definitively limiting its maximum height and overall biomass accumulation. The laws of physics ultimately dictate the absolute ceiling. So the height of the tallest redwood isn't limited by its genetics or how old it gets or the soil it lives in. It is strictly limited by the sheer physical impossibility of sucking water up any higher without boiling it inside the trunk. That is phenomenal.
It really reframes how you look at the canopy.
All right, we've spent a massive amount of time talking about how water goes up, but a tree also has to move things down. It manufactures all those sugars in the high canopy, and it has to somehow feed those living mining roots deep underground in the dark.
And that requires a completely different, entirely separate vascular system. Welcome to the bizarre biology of the phloem.
The phloem is arguably even more fascinating than the xylem. While the xylem utilizes dead, empty tubes and physical vacuums to pull water upward, the phloem faces a different challenge. It must actively transport photosynthetically derived sugars, complex amino acids, and vital hormonal signaling molecules downward and outward to the rest of the massive organism.
And to accomplish this active transport, the phloem utilizes what must be considered one of the most bizarre, extreme cellular states in the natural world cells, existing perpetually as the living dead.
The living dead. The zombie cells of the tree. Let's really unpack this because it's so strange. The primary conducting cell in the phloem network is called a sieve element. Now, when a sieve element is newly born in the cambium, it's a completely normal, fully functional plant cell, right? It has a nucleus, DNA, the whole package.
Yes, initially.
How does it transition into a zombie?
During its maturation process, which is called phloemogenesis, the sieve element undergoes a highly selective, terrifyingly precise form of partial autophagy. Similar to the xylem, it urgently needs to clear its internal physical space to eliminate fluid resistance to the flow of thick, sugary sap.
So it systematically begins to digest its own internal machinery. It completely dissolves its own nucleus, erasing its genetic code. It destroys its central vacuole, its Golgi apparatus, its ribosomes, and it dismantles its entire structural cytoskeleton.
It literally dissolves its own brain and its own vital organs.
But earlier, we talked about the xylem doing this exact same thing, and the xylem cells die completely. They just become empty, dead pipes. How is the sieve element different? How is it a zombie?
It is fundamentally different because it miraculously arrests this degradation process just moments before total cellular death. The sieve element manages to retain a functioning plasmalemma, which is the vital outer cell membrane, maintaining a highly pressurized living boundary against the outside world.
And it also carefully keeps a heavily modified, smooth endoplasmic reticulum, right?
Yes, which it presses flat against the inner cell periphery to act as a localized storage unit for calcium ions and specific phloem proteins. And it retains just a few rudimentary, barely functional plastids and mitochondria.
So it's not truly dead because it maintains a pressurized membrane, but it has absolutely no nucleus, no DNA, and no ribosomes to make new proteins. It is completely brainless, hollowed out, and genetically silent. It's nothing but a pressurized biological tool. But how does a cell with no nucleus or ribosomes actually survive for months or years? How does it repair its membrane if it gets damaged? How does it manage its own complex metabolism? It literally can't do anything for itself.
Left alone, it cannot survive independently for more than a few hours. It is entirely, hopelessly sustained by its ontogenetic sister cell. When the cambial stem cell originally divided to create the sieve element, it simultaneously created an adjacent, intimately linked companion cell.
And these two specific cells share a profound, absolutely obligatory metabolic codependence. I know there's a good analogy for this relationship.
Right, you had one earlier that fit perfectly.
Yeah, I think the best way to visualize this is to compare it to a deep sea diver on remote life support. The
Yeah.
sieve element is the hollow, brainless diver down in the dark, doing the heavy physical work of conducting the pressurized sap. But the diver has no air tanks, no food, no ability to think.
They are connected by a critical umbilical cord to a heavily equipped support ship floating on the surface, which is the companion cell.
Exactly. The companion cell has the massive nucleus, the ribosomes, the genetic blueprints, the energy production. It actively pumps everything the diver needs down that umbilical cord to keep them alive and functioning while they work in the pipe.
That is an exceptionally accurate functional analogy. At the cellular microscopic level, that vital umbilical cord consists of highly specialized structures known as pore plasmodesmata units connecting the two adjacent cells.
These are relatively massive, heavily regulated channels, right?
Yes, they allow the continuous passage of large macromolecules up to 70 kilodaltons in size directly from the companion cell's cytoplasm into the sieve element.
70 kilodaltons is absolutely huge in molecular biology terms. That means entire, fully folded large proteins are squeezing through those pores.
Yes, they have to. The companion cell operates completely as the sieve element's remote life support system and metabolic engine. Because the sieve element lacks ribosomes, it cannot synthesize a single protein to repair its own membrane.
So the companion cell constantly reads the DNA, synthesizes all the required phloem-specific proteins, such as structural P proteins, and actively, continuously pumps them through those massive pores into the sieve tube to maintain cellular integrity.
And immediately plug leaks if the highly pressurized phloem is ever damaged by an insect.
It's an incredible, extreme division of labor. One gives up its brain to become a pipe, the other gives up its mobility to become a life support system. But there is a very recent super counterintuitive discovery about how these companion cells actually get the massive amounts of energy required to do all this relentless pumping, right? It has something to do with their chloroplasts.
Yes, this is a truly fascinating area of recent molecular research that upends basic assumptions. In introductory biology, we are always taught that chloroplasts, the green organelles inside plant cells, have one single primary job: capturing carbon dioxide from the atmosphere and fixing it into sugars via the process of photosynthesis.
However, recent, highly detailed tissue-scale flux balance analyses have demonstrated that the chloroplasts residing deep within these companion cells operate entirely differently than the standard chloroplasts found up in the leaf mesophyll.
Yes, they do.
They aren't capturing carbon to make sugar. What on earth are they doing with the light?
They experience what we might term a severe metabolic repurposing. Rather than fixing carbon from the atmosphere, which is energetically expensive, the companion cell chloroplasts function primarily to provide photosynthetically generated ATP.
ATP is pure, usable cellular energy.
Right. They act as highly localized, hyper-efficient power plants. They burn the captured light energy purely to generate the massive amounts of ATP required to constantly energize the plasma membrane proton pumps. These specific proton pumps are what physically drive the active, energy-intensive loading of sugars from the leaves into the highly pressurized phloem network.
So the chloroplasts inside the companion cell completely abandon their role as sugar factories and just become massive, dedicated batteries to power the pumps. This entire system, one cell sacrifices its autonomy to become a hollow pressurized pipe. The other serves as its remote brain and dedicated solar battery. It represents an absolute peak of evolutionary refinement and multicellular cooperation.
It truly is.
So we've covered the internal plumbing. The suicidal xylem moving water up through physics, the zombie phloem moving sugars down through active pumping. All of this incredible biological machinery is wrapped up in the solid material we simply call wood. And I think we heavily take wood for granted. We chop it up, we burn it in fireplaces, we build cheap disposable furniture out of it.
We really do.
But from a pure engineering standpoint, wood is actually a phenomenal super material, isn't it?
It is an extraordinary, highly optimized biological metamaterial. The immense evolutionary success of trees, their ability to grow to such towering heights, is inextricably linked to the precise biomechanics of wood. If we zoom out from the fluid dynamics of the individual cells, look at wood at the microstructural material level, it is a highly sophisticated, hierarchical nanocomposite.
A hierarchical nanocomposite. Let's break that technical term down for everyone listening. What are the specific structural components that make it a composite?
It consists primarily of highly ordered crystalline cellulose microfibrils. These are incredibly strong microscopic threads, and they are embedded firmly within a complex, amorphous, glue-like matrix composed of hemicellulose and the highly cross-linked lignin we discussed earlier.
The cellulose microfibrils are wound helically, like a spring, through the cell wall. They provide immense tensile strength. As we established, they act as the biological rebar.
So they aggressively resist being pulled apart or stretched by the wind.
Correct. And the surrounding dense lignin matrix provides the vital compressive stiffness, the ability to resist the crushing downward weight of the thousands of pounds of tree above it, as well as essential shear resistance and total waterproofing.
The lignin effectively binds and locks the tensile cellulose elements together into a cohesive, unyielding whole. When you actually compare this biological biocomposite to our modern, heavily engineered construction materials, the performance metrics are absolutely staggering.
Particularly when we look at a specific engineering metric called specific strength.
Exactly.
Specific strength, that's essentially the strength to weight ratio, right? How strong a material is relative to how heavy it is to move or support.
It is a critical metric for any form of structural engineering, whether you are trying to build a 300-foot tree or a modern commercial airplane. Let's look at some comparative statistics to ground this. High-quality structural timber, when loaded parallel to its internal grain, exhibits a compressive strength of roughly 40 to 70 megapascals and a robust tensile strength of 80 to 120 megapascals.
Okay, those are just numbers to me. How does 70 megapascals compare to, say, the concrete we use to build our massive city skylines?
Non-reinforced concrete, standard C25 or C30 grade, used in typical construction, has a compressive strength of maybe 25 to 30 megapascals, which is lower than wood, and has an exceptionally poor, almost negligible tensile strength of only 2 to 5 megapascals.
That extreme weakness and tension is precisely why concrete absolutely requires internal steel rebar reinforcement. Otherwise, it cracks and snaps under any bending tension.
Furthermore, concrete is extremely heavy and densely packed. Consequently, when you divide its strength by its weight, concrete has the lowest specific strength among all primary construction materials. Wood is vastly, demonstrably, structurally superior in terms of pure efficiency. The specific structural efficiency of wood is actually 4 to 5 times higher than non-reinforced concrete.
So wood absolutely crushes concrete in a pound-for-pound structural fight. What about steel? We think of steel as the ultimate building material.
Structural steel, like standard ASTM A36 used in I-beams, features very high absolute strength, around 250 to 500 megapascals in both compression and tension, and it has unmatched shear resistance and ductility. However, steel is extraordinarily dense. It is incredibly heavy material.
As a result, when you calculate its specific strength to weight efficiency is actually lower than that of high-quality, lightweight wood.
It really is.
That is simply amazing. Pound for pound, a piece of natural structural timber is more efficient at holding a load than a heavily engineered beam of solid structural steel. The only thing that really beats it is advanced carbon fiber composites synthesized in labs, which are over 1,000 megapascals, but those are insanely expensive and highly energy intensive to manufacture. The tree simply grows its incredible composite out of raw dirt and air using ambient sunlight.
And remarkably, modern human engineering is now attempting to reverse engineer and actively replicate the performance of those advanced carbon composites, using humble wood as the base material. Material scientists have recently developed a complex process to create what is colloquially known in the literature as superwood.
Superwood. How exactly do you take natural wood and make it super?
They utilize a highly specific heated chemical bath to carefully dissolve and entirely remove the lignin and hemicellulose matrix from the natural wood block, leaving behind primarily the porous structure of cellulose microfibrils.
Then they subject this weakened, porous cellulose structure to intense mechanical and thermal compression. They literally crush it flat under high heat and immense pressure.
Exactly.
So you chemically remove the concrete and then you physically crush the rebar tightly together. What does that actually achieve materially?
It forces the remaining highly aligned cellulose nanofibers to tightly pack together, eliminating all the microscopic empty space, and allows them to form incredibly dense, overlapping hydrogen bonds with each other.
This engineered, forced densification results in a final material that boasts a specific tensile strength greater than most modern metal alloys. In fact, it exhibits impact resistance and ballistic absorption capabilities that are rapidly approaching those of Kevlar armor.
We are literally turning blocks of wood into Kevlar-like armor by mimicking and intensifying its natural nanostructure. That is wild.
But wood isn't just a strong building material. As we touched on earlier when we discussed tree rings, it's also a high-fidelity archive. The annual deposition of these microstructures permanently encodes high-resolution environmental data.
Yes, the archival capacity of wood is unparalleled in biology because cambial cellular activity fluctuates continuously with seasonal moisture, temperature variations, and ecological stress, the physical width, the cellular density, and crucially, the specific isotopic signature of each individual tree ring provides a flawless, chronologically precise environmental archive.
Through the exacting science of dendrochronology, researchers can extract a highly precise climate history. They can track centuries of specific, localized drought stress events. They can identify years with global volcanic winters caused by distant, massive eruptions. And they can precisely map historical ecological disturbances like wildfires or insect outbreaks, all perfectly embedded directly within the biological matrix of the organism.
The wood is quite literally passively recording the atmospheric history of the Earth.
It is awe-inspiring to think about. So we have this incredible, towering, data recording, super material internal structure. But we have to remember, it is completely full of massive quantities of stored chefs in the phloem and fresh water in the xylem.
Just a huge target.
In a hungry, thirsty, highly competitive forest ecosystem, that trunk is essentially a giant, unprotected buffet for every insect and fungus around. It urgently needs armor. Which brings us to the outer defenses. Let's talk about bark.
The bark is the tree's primary dynamic interface with a highly hostile external environment. And, much like the inner wood, it is actively and continuously generated by a dedicated secondary lateral meristem. While the vascular cambium builds the inner wood and phloem, the tough outer bark is generated by a completely separate layer called the phelogen, which is more commonly known as the cork cambium.
So there's a second, completely separate microscopic cylinder of stem cells out near the surface of the trunk.
Yes. And exactly like the internal vascular cambium, the outer phelogen divides bifacially. It produces living parenchyma cells toward the inside, known as the pheloderm, which support the active metabolic functions of the bark. And it produces phellem, or cork cells, toward the outside.
And these cork cells, the phellem, they are the physical armor. What makes a cork cell so exceptionally tough?
As these outer phellem cells mature and are continually pushed outward toward the surface, they undergo a highly specialized form of programmed cell death. But before they die and become part of the outer shield, they heavily, thickly impregnate their primary cell walls with a remarkable substance called suberin.
Suberin is a highly complex, extremely hydrophobic aliphatic network composed of long-chain fatty acids, dense alcohols, and aromatics.
Yes.
Hydrophobic meaning it aggressively repels water.
Violently repels water. Once those outer cork cells finally die and leave behind this interlocking microscopic matrix of suberin-soaked walls, it forms a virtually impenetrable physical barrier. It is an absolute hermetic seal against the outside world. It prevents lethal desiccation, preventing the tree's internal water from evaporating through the trunk, and physically blocks the entry of fungal and bacterial pathogens.
However, a completely airtight total seal would quickly suffocate the living, respiring phloem and cambium cells trapped beneath it. To solve this engineering paradox, the bark features specialized, highly porous respiratory channels called lenticels, which permit controlled, strictly necessary gas exchange while maintaining the armor.
So it's essentially wearing a waterproof, highly durable Kevlar vest equipped with little microscopic breathing vents.
But bark isn't just passive physical armor like a stone wall. You described it as a dynamic, highly reactive immune system. What actually happens when something violently breaches the wall? When a deer scrapes its antlers against the bark, or a bear claws it deep enough to expose the wood?
When physical trauma severely breaches the periderm, the tough outer bark layers, the tree does not passively bleed. It initiates a rapid, highly localized, aggressive wound response. Unspecialized living parenchyma cells located deep in the underlying pheloderm or phloem immediately detect the chemical signals of the damage.
They rapidly undergo a process called de-differentiation. They literally revert their genetic state from mature, specialized cells back to a flexible, highly proliferative stem cell-like state.
They realize the castle wall is breached, and they immediately turn back into raw stem cells to build a patch.
Exactly. They organize and form a brand new, deeper, localized layer of phelogen, a temporary emergency cork cambium directly beneath the site of the wound. This new, localized cambium quickly generates a fresh, thick layer of highly suberized cork to seal the breach from behind, effectively and permanently isolating the damaged, exposed, potentially infected tissue from the healthy interior. It's a highly effective biological quarantine mechanism.
But the tree doesn't just quietly patch the hole and hide, it fights back aggressively. The chemical warfare deployed by trees is honestly terrifying when you look closely.
We always anthropomorphize plants as helpless, passive victims, but they are absolutely vicious at the biochemical level.
They are heavily armed, and they do not hesitate to deploy those weapons. Simultaneously with the physical suber and wound healing, the bark and phloem rapidly deploy an extensive, complex arsenal of both constitutive meaning always present and induced chemical defenses. The tree possesses specialized anatomical structures like resin ducts, laticifers, and defensive parenchyma cells that constantly synthesize a vast array of toxic secondary metabolites.
We are talking about highly toxic terpenoids, complex, heavy tannins, and potent neurologically active alkaloids. These compounds function as highly sophisticated, targeted biological weapons.
Let's focus on cannins for a second, because humans actually consume those in red wine and black tea. We like the bitter taste. But to an invading insect, a tannin is a literal nightmare.
To a herbivorous insect attempting to eat the tree, yes, it is lethal. Tannins act as powerful, highly disruptive anti-nutritive agents. When a caterpillar ingests bark or leaves laden with high concentrations of tannins, those complex molecules chemically bind to and immediately precipitate the vital digestive enzymes within the insect's own gut.
They literally solidify and deactivate the insect's digestive proteins. The insect can continue to voraciously eat the leaves, but it can no longer extract a single calorie of nutrition. It functionally starves to death with a completely full stomach.
It starves them from the inside out while they are eating. That is diabolical. And what about the sticky stuff? The thick resins that pine trees aggressively bleed when you cut into them with an axe.
Those sticky, highly viscous, terpene-rich resins are dual-purpose weapons. They are both severe chemical toxins and inescapable physical traps. When a boring beetle chews deep into the bark, the tree detects the vibration and chemical signature, and it responds by rapidly exuding this viscous resin under high hydrostatic pressure directly into the insect's newly chewed wound canal.
The resin contains highly toxic, volatile compounds that immediately poison the beetle's nervous system. And simultaneously, the sticky mass physically entombs the insect, completely encapsulating it and literally flushing its suffocating body out of the injury site.
The tree actively bleeds toxic glue to drown and flush out its attackers. It is absolutely brutal.
And this complex, armored, heavily defended outer layer is so incredibly vital to the organism that disrupting it is rapidly fatal. This perfectly explains why girdling a tree kills it so effectively, right?
Yes, the lethal absolute nature of girdling or ring stripping perfectly demonstrates the absolute, unbroken interdependency of the tree's dual vascular systems. If you take an axe and remove a complete circumferential strip of bark all the way around the trunk, down just to the depth of the cambium, you will notice the tree does not die immediately.
The inner xylem, the deep wood, remains perfectly intact, so the cohesion tension mechanism continues pulling water up to the canopy. The leaves stay bright green. The tree appears completely fine and healthy for months.
But it's a dead tree walking. Why does it eventually die if it has water?
Because by removing that ring of bark, you have physically, permanently severed the downward phloem pipeline. The leaves high in the canopy are still happily, continuously producing photosynthetically derived sugars, but those vital sugars hit the cut in the bark and can travel no further downward. They pool at the cut.
Because the massive subterranean root system is now instantly, permanently cut off from its only food supply, it has to rely entirely on whatever stored starch reserves it happens to have sequestered in its tissues.
And what happens when those hungry roots finally run out of stored food?
They slowly, inevitably starve to death in the dark. Eventually, the subterranean root system exhausts its metabolic reserves and cellular death cascades through the roots. And once the root tips die, they immediately stop actively absorbing water from the soil.
That sudden lack of incoming water snaps the tension and halts the upward transport of water through the intact xylem. The canopy finally wilts, and this cascading failure leads to the total systemic collapse and death of the entire massive organism, all because of a single, shallow, severed ring of bark.
It really heavily emphasizes that the massive visible canopy we admire is utterly, hopelessly reliant on the invisible root system we walk on. Speaking of which, let's finally go underground. Because roots don't just sit there passively in the dark drinking water like a kitchen sponge. The subterranean engine of a tree is violently biochemically active. They're basically engaging in aggressive chemical mining operations.
That is a highly accurate, functionally apt description. The monumental visible architecture of a tree is entirely dependent on the complex, aggressive biogeochemical engineering conducted continuously by its root system deep in the lithosphere. First, we must understand the fundamental mechanism of root osmotics. Roots do not just wait for water, they actively manipulate the chemical conditions of the soil immediately surrounding them, an area called the rhizosphere.
They utilize massive amounts of cellular energy ATP to actively transport mineral nutrient ion salts out of the surrounding soil water and across their cell membranes into the inner root xylem.
Oh, so they intentionally pump salts into themselves to make themselves salty.
Yes, and by highly concentrating these mineral ions inside the root cylinder, they establish a powerful, localized osmotic potential gradient. The laws of thermodynamics dictate that water naturally wants to flow from an area of low solute concentration to high solute concentration to achieve equilibrium. This severe gradient forcibly, continuously draws water from the surrounding soil into the root, generating a positive internal hydrostatic pressure known as root pressure.
Now, while this positive root pressure can independently push sap upward by several meters, which you sometimes observe as water droplets forming on leaf tips at night, a process called guttation, it is vastly insufficient to push water 100 meters to the high canopy. That requires the massive cohesion tension mechanism we discussed earlier.
Right, the root pressure is just a tiny primer pump at the bottom. The sun provides the main, massive pull from the top. But absorbing water is only half the battle down there. The roots urgently need essential minerals to build their enzymes and cell walls. They need phosphorus, they need calcium, they need magnesium.
But in many dense rocky forests, those nutrients aren't just floating freely in the soil water, they are locked up completely solid inside the crystalline matrix of rocks. How does a soft, fleshy, microscopic root tip get solid calcium out of a piece of solid granite?
To acquire those essential, highly recalcitrant nutrients locked within solid rock matrices, tree roots engage in aggressive, active chemical weathering. They effectively mine the solid soil. The roots, heavily concentrated at the actively growing microscopic root tips, continuously exude substantial quantities of low molecular weight organic acids directly into the surrounding soil matrix.
The principal acids utilized in this biological mining are citric acid, oxalic acid, and malic acid.
They literally sweat harsh acids into the dirt.
Yes, and these exuded organic acids function as powerful chemical chelating agents. When exuded into the microscopic soil pores, they chemically bind directly to the mineral surfaces of the soil particles and solid bedrock. They physically and chemically dissolve the solid rock matrix, forcefully ripping the mineral bonds apart.
This intense, localized biological weathering significantly increases the solubility and availability of highly recalcitrant nutrients like phosphorus and essential metallications, freeing them up to be immediately absorbed by the waiting root network.
So the roots are quite literally chemically dissolving the deep geological foundations of the earth to build their towering trunks. It's incredible. But pouring all these intense organic acids and simple sugars into the soil has a highly bizarre, very important side effect. It creates this massive counterintuitive paradox regarding global soil carbon, doesn't it?
It creates a deeply counterintuitive, highly complex twist in our understanding of global soil carbon dynamics. For a very long time, ecologists and climate scientists operated on a logical assumption that these dense root exudates simply added carbon to the soil matrix. A tree continuously pumps carbon-rich sugars and acids into the dirt. Therefore, the dirt must get richer in stored carbon.
It seems perfectly logical. However, recent, highly sophisticated biogeochemical research utilizing radioactive isotope tracking has revealed a deeply paradoxical priming effect.
What actually happens when the tree pumps all those simple, easily digestible sugars and acids into the dirt?
It acts like highly combustible high-octane fuel for the massive soil microbiome. The sudden influx of these easily digestible exudates stimulates intense, localized, explosive microbial activity. The billions of bacteria and fungi in the soil essentially go into a hyperactive feeding frenzy.
And these microbes, entirely supercharged by the tree's exudates, don't just lazily eat the simple sugars provided. Their massively increased metabolic activity allows them to aggressively attack and accelerate the decomposition of highly stable, mineral-associated organic matter that had previously been safely locked away in the soil for centuries.
Wait, let me make sure I understand this. So the microbes get hyped up on the free tree sugar, and then because they are so energized, they turn around and burn down the ancient, stable carbon reserves in the soil.
Exactly. By dramatically stimulating the local microbiome, the tree's root exudates can actually measurably lead to an unexpected net loss of long-cycling, deep soil carbon as the hyperactive microbes rapidly respire it back into the atmosphere as CO2 gas.
It is a brilliant, sobering example of exactly how complex, nonlinear, and unpredictable these massive biogeochemical feedback loops can be when we try to model them.
That is absolutely wild. The tree furiously builds its massive carbon body above ground, but it might actually be actively burning ancient soil carbon below ground to do it.
Okay, before we leave the deep roots, I have to talk about a phenomenon called hydraulic redistribution. Because earlier we spent a lot of time talking about how water moves aggressively up during the hot day. But what happens at night? The roots do something that seems totally, completely backward.
They exhibit complex, highly counterintuitive behavioral hydraulics that completely defy our simple, unidirectional upward flow models. Consider the cycle. During the night, the sun sets. The temperature drops. The stomata in the high leaves clamp shut to conserve water. The intense, solar-driven transpirational pull from the canopy ceases entirely.
The massive vacuum pump at the top turns off, so the water in the tree just sits there statically, waiting for morning.
Not at all. The extreme water potential gradients that built up during the hot, stressful day within the vast, sprawling root system begin to aggressively seek physical equilibrium. Consider a large, mature tree growing in an arid environment. It has massive, deep tap roots that penetrate far down into moist, subterranean aquifers, and it also possesses a wide, sprawling network of very shallow, fine roots located in the bone-dry upper topsoil.
During the night, while the canopy is resting, those deep tap roots continue to passively absorb water from the wet aquifer.
But the canopy isn't pulling it up anymore. The vacuum is gone. So where does that deep water physically go?
It follows the exact path of least physical resistance. Rather than pushing it all the way up the immense, heavy trunk against gravity, the water moves laterally and actually flows downward and outward along the localized water potential gradient within the root system itself. It flows continuously from the deep, wet tap roots up into the central root crown and then out into the completely dry, shallow, fine roots.
And crucially, it actually leaks out of those fine roots into the surrounding dry soil.
The tree is literally watering the dirt. It pulls water from the deep hidden aquifer and deliberately, actively leaks it into the dry surface soil. Why on earth would a plant in a dry environment give up its hard-earned water to the dirt?
This fascinating phenomenon is known as hydraulic redistribution, or more specifically, hydraulic lift. The tree is effectively deliberately irrigating its own superficial fine roots during the night. You see, the shallow topsoil roots are where the vast majority of critical nutrient absorption takes place. That's where the decaying organic matter is. But those delicate fine roots die very quickly if the topsoil becomes totally desiccated.
By actively transferring water from deep, wet layers to shallow, dry layers every single night, the tree guarantees the survival and viability of its critical upper absorptive root network.
It's spending water to keep its primary mining equipment alive so it can get phosphorus tomorrow.
Exactly. And incidentally, this nightly irrigation also inadvertently sustains the surrounding highly beneficial microbial community in the topsoil, fundamentally enhancing overall ecosystem productivity and resilience during severe summer droughts. It is a masterful, deeply evolved manipulation of local hydrology.
Okay, so we've firmly established that roots are active, mining, acid-sweating, self-irrigating marvels, and they don't operate down there alone. They partner with fungi. Which brings us to one of the most fascinating, famous, and currently most hotly debated biological theories of the last 20 years, the mother tree hypothesis.
Yes, this is a critical topic to cover. To begin with the established science. It is a widely established, completely undisputed biological fact that the vast majority of tree roots globally form intimate symbiotic relationships with mycorrhizal fungi. The microscopic fungal hyphae effectively extend the root system's physical absorptive surface area, forging aggressively through microscopic soil pores that the larger plant roots simply cannot reach.
They actively scavenge and trade these critical soil nutrients, primarily nitrogen and phosphorus, back to the tree in direct exchange for plant-derived photosynthetic carbon sugars. It is a highly effective, strictly transactional mutualism.
Right, that basic transactional symbiosis is textbook biology. But over the last couple of decades, this idea heavily evolved in the scientific literature and in the media into something much grander. It gave rise to the highly popularized mother tree hypothesis. This is the narrative that absolutely captured the public imagination. The comforting idea that forests are essentially massive socialist networks.
The theory posits that massive, mature overstory trees act as central, highly altruistic hubs in a vast, interconnected, common mycorrhizal network.
The central proposition of this hypothesis is that these older established trees actively and deliberately share massive amounts of carbon, vital nutrients, and even chemical distress signals directly through the fungal pipelines with vulnerable, unrelated seedlings struggling in the shaded understory. The heavy implication is that the mother tree is actively nurturing its offspring and kin, deliberately ensuring their survival in a harsh, competitive environment.
It's a beautiful, deeply comforting story. It paints the forest as a caring, interconnected family. But science isn't always about what is comforting. And as impartial observers of the data, we have to look at how the broader scientific community has responded to this specific hypothesis recently. Because there's been a massive, fierce, heavily peer-reviewed critique of this hypothesis in the last few years, hasn't there?
There has been a profound, highly rigorous scientific reevaluation. The scientific pushback from ecologists has been intense, fundamentally challenging these older, highly anthropomorphized models. Extensive, meticulous re-examinations of the underlying empirical data, most notably a highly comprehensive 2023 meta-analysis published by Karst and colleagues, alongside deeply critical reviews by Henriksen and others, strongly indicate that the empirical evidence for actual altruistic resource sharing via fungal networks is highly limited, methodologically fragile, and frequently plagued by severe citation bias within the popular literature.
Let's break down the specific methodological scientific critiques here, because it's important. First, how are the original researchers measuring this supposed carbon transfer in the first place? And what exactly is the structural flaw in that measurement?
The foundational evidence for direct carbon transfer from a mother tree to a shaded seedling relies heavily on tracking carbon isotopes. Researchers expose the large canopy tree to a rare, easily traceable isotope like carbon-13 or radioactive carbon-14, and then days later, they look for trace amounts of that specific isotope appearing in the nearby understory seedlings.
And they did find the isotope in the seedlings, right? So doesn't that inherently prove the fungal pipeline works as a delivery system?
It rigorously proves that the isotope physically moved from point A to point B, but it completely fails to prove how it moved. The critical fatal flaw in the methodology is that these recorded isotopic shifts are often minuscule, frequently operating at the very statistical limits of detection. And these tiny transfers completely fail to differentiate between a direct, active fungal pipeline and alternative, purely physical pathways in the soil.
For instance, the large tree naturally constantly exudes heavy amounts of isotopic root, exudates those organic acids and sugars directly into the soil water, which passively diffuses through the dirt and is naturally taken up by the seedling's own roots. Or, even more simply, the billions of soil microbes rapidly respire the big tree's isotopic carbon back out, as CO2 gas, which floats up from the dirt and is immediately absorbed by the seedling's leaves via standard daytime photosynthesis.
So the little seedling might just be passively breathing the radioactive exhaust of the big tree, not receiving a deliberate care package through a dedicated fungal tube.
Precisely. And highly damaging to the pipeline hypothesis, rigorous, isolated control studies have frequently shown that the rate of isotopic transfer between plants remains statistically identical, regardless of whether the physical fungal-hyphal connections are left perfectly intact or intentionally physically severed by a microscopic mesh. If physically cutting the wire doesn't stop the signal, the wire wasn't carrying the signal.
That is a devastating methodological critique of the data. But there's also a deeper theoretical problem you mentioned earlier. You called it evolutionary incongruence. Why doesn't it make fundamental evolutionary sense for a fungus to act as a socialist delivery service for a forest?
We must remember the fundamental nature of the organism. Mycorrhizal fungi are not passive, altruistic, plant-loving conduits. They are independent, highly competitive, distinct biological organisms driven entirely by natural selection to aggressively maximize their own evolutionary fitness. Fungi operate on a strict, energetic exchange rate. They trade hard-to-find nutrients for easy carbon. It makes absolutely no evolutionary or energetic sense for a fungus to actively tax a massive, highly productive, carbon-rich host tree, which is an excellent, reliable resource, merely to subsidize and energetically feed a dying, heavily shade-suppressed seedling that offers absolutely no metabolic return on investment. The fungus works strictly for the highest bidder. It does not run a forest charity.
It's a harsh biological reality. And finally, what happens when we zoom out and look at the actual long-term field data? If mother trees are actively nurturing seedlings, we should theoretically see lots of healthy, thriving seedlings clustered tightly around the base of the big trees, right? What do the actual massive forest studies show?
The extensive field data frequently directly contradicts the core premise of the hypothesis. Long-term data from dense boreal forests, consistently demonstrate that large overstory trees intensely suppress, rather than facilitate, seedling establishment. Forests are, at their core, zones of intense, ruthless, below-ground competition for highly limited water and mineral nutrients. Seedlings attempting to grow within the active root zone of massive mature trees exhibit significantly reduced growth rates and dramatically higher mortality.
Do we have specific data points or numbers on that suppression?
Yes. For example, highly controlled, extensive studies conducted in Scandinavian pine stands reveal a stark, competitive reality. Young trees attempting to grow within a 5-meter radius of a retained, mature overstory tree suffered a severe, measurable 24% reduction in total biomass growth, compared to identical seedlings growing further away in open, untethered soil. The mature tree isn't actively feeding them. Its massive, highly efficient root system is actively starving them of essential water and minerals.
A 24% growth penalty just for living near the mother tree. That is ruthless competition.
It is. Now, this is absolutely not to say that trees don't interact dynamically. Localized chemical signaling and trace elemental transfer undoubtedly occur in the highly complex rhizosphere. However, the sweeping, popularized assertion that entire forests operate as highly cooperative, socialist networks via intentional fungal pipelines severely oversimplifies and largely misrepresents the fierce, highly competitive Darwinian reality of forest ecology.
It's a truly fascinating debate, and it serves as a great reminder that attempting to apply human morality, like altruism or charity, to ancient biological systems usually leads us astray. But despite this fierce, ruthless competition for light and water, some of these individual organisms manage to win the evolutionary game so thoroughly that they essentially break the biological clock entirely. Which brings us to perhaps the most mind-bending concept: biological near immortality.
This is perhaps the most profound physiological distinction between trees and animals. The vast majority of animals, including humans, undergo an inescapable process called genetically programmed somatic senescence. This means we have a predictable, intrinsic physiological decline tightly built into our very DNA that leads inevitably to aging, systemic failure, and death.
Right, a dog lives maybe 15 years, a human lives roughly 80. Our bodies just physically wear out on a microscopic cellular level over time, but trees simply don't do this.
No, they exhibit a phenomenon termed negligible senescence. Because, as we established way back in the beginning, the vast majority of a mature tree's massive biomass is already entirely dead, and its incredibly tiny fraction of living tissues are continually, endlessly regenerated by immortal meristems located in the cambium and the root tips. Trees simply do not age in the conventional organismal sense. Theoretically, the cellular machinery of a tree's stem cells is capable of infinite, endless self-renewal.
Infinite self-renewal, but how is that possible? What is the specific molecular secret? Because in human cells, we have this ticking genetic clock called telomeres that enforce our mortality.
Yep.
Let's explain exactly what those are to understand how trees beat them.
At the deep molecular level, aging in animals is largely driven by the continuous, unavoidable attrition of telomeres. Telomeres are protective nucleoprotein caps located at the very ends of linear chromosomes. Think of them exactly like the small plastic aglets at the end of a shoelace that physically stop the shoelace from fraying and unraveling. With every successive necessary cell division, these telomere caps physically shorten just a little bit. When they finally reach a critical, minimum threshold length, it violently triggers massive DNA damage responses. The cell permanently stops dividing, enters replicative senescence, and the overall tissue visibly ages.
But trees completely bypass this biological countdown. How do they keep their genetic shoelaces from fraying for 5,000 years of continuous cell division?
Exhaustive molecular research on incredibly ancient specimens has finally revealed their secret. Look at the famous bristlecone pine, Pinus longiva, which is known to live for over 5,000 years, or the millennial Ginkgo biloba. Researchers have discovered that these ancient organisms maintain highly active telomerase enzymes continuously throughout their immense lifespans. Telomerase is the specialized enzyme that actively rebuilds telomeres, but it goes significantly beyond just having the enzyme present. They have highly specialized structural proteins supporting it.
Let's talk about the major ginkgo discovery. What exactly did scientists find inside the cells of Ginkgo biloba?
In Ginkgo biloba, molecular scientists identified a highly specialized double-stranded telomere-binding protein, officially designated as GBTRB1. This specific protein actively interacts directly with the telomerase subunit. It essentially acts as an unbreakable molecular stabilizer, ensuring the absolute structural integrity of the telomere complex, even in incredibly old, highly mature tissues. As a direct result of these mechanisms, the telomere lengths in these ancient trees simply do not undergo the linear terminal shortening seen in all animals.
So they just stay the exact same length forever.
Better than that, actually. They display a highly dynamic, complex, cyclical pattern of lengthening and shortening based strictly on seasonal growth phases. And remarkably, in some cases, empirical measurements show that the average telomere length actually increases as the tree ages. Think about what that implies. The vascular cambium of a 5,000-year-old bristlecone pine remains perpetually molecularly youthful. It maintains the exact same vigorous, flawless regenerative capacity as it had when the organism was just a 50-year-old sapling.
The cells are literally younger at a fundamental molecular level than they were 1,000 years ago. That is staggering to contemplate, and this negligible senescence leads to biological records that just completely shatter the human perception of time. A bristlecone pine living 5,000 continuous years is insane, but clonal colonies push this to a geographical scale that is almost terrifying. Let's talk about Pando.
Pando is the ultimate massive expression of this biological immortality. Located in Utah, Pando is a massive clonal colony of a single male quaking aspen. Above ground, to the untrained eye, it appears to be a massive sprawling forest consisting of over 40,000 individual distinct tree trunks. However, rigorous genetic testing reveals they are all completely genetically identical. They are all physically connected by a single massive, unified subterranean root system.
So it's not a forest at all. It's a single, continuous, highly distributed organism with 40,000 heads. How old is this single organism estimated to be?
Because it continually, relentlessly generates new trunks from its roots while older trunks die and fall, the continuous, unbroken biological existence of the massive Pando root system spans tens of thousands of years. It has outlived multiple ice ages, it predates human civilizations, and it has survived massive, sweeping climatic shifts. It is an organism that has essentially conquered the concept of time.
But nothing lives forever in reality. If a massive tree does not die of old age, if its DNA never degrades and its telomeres stay long and perfect, what actually finally kills a 5,000-year-old tree?
The inevitable, unavoidable fall. If they do not die of an endogenous, internal biological clock, they are ultimately invariably killed by the external environment and the absolutely unforgiving laws of physics. The ultimate limit on a non-clonal tree's longevity is governed by what ecologists call the growth-decay dichotomy.
The growth-decay dichotomy. Let's really explore the physics of this stuff.
As a tree, perpetually, inevitably adds massive amounts of mass and height year after year, two specific physical burdens increase relentlessly. First, the pure mechanical load on its massive trunk and lower structural branches becomes staggeringly heavy. It is fighting a constant, exhausting, centuries-long battle against gravity. Second, the internal hydraulic path length, the sheer physical distance from the deepest, wettest roots to the highest transpiring canopy, becomes dangerously energetically extended.
It just takes more and more and more energy to pull the water higher and higher as it grows.
Yes, and concurrently, the immense energetic cost of maintaining that ever-expanding, massive three-dimensional network of living tissue, particularly those thousands of living ray parenchyma cells deep in the sapwood that we discussed earlier, eventually begins to severely outstrip the total photosynthetic output of the canopy. The tree simply becomes metabolically strained just trying to maintain its own enormous bloated infrastructure.
It's like a massive, sprawling empire that has expanded too far, stretched its supply lines too thin, and can no longer afford to pay its army or fix its crumbling roads. What actually happens when it finally hits that critical metabolic tipping point?
At this critical threshold, the tree's incredible, age-defying modular growth capacity is finally exceeded by its sheer physical limitations. Because it is chronically energy-starved, the organism becomes incapable of reacting fast enough to threats. It cannot generate enough metabolic energy to rapidly seal new bark wounds with suberin. It lacks the deep metabolic reserves to synthesize the massive amounts of toxic tannins and lignins necessary to compartmentalize and fight off fungal decay entering through broken branches.
So its active immune system and its structural armor finally failed because it simply ran out of power.
Precisely. Therefore, death in ancient trees is invariably the result of an exogenous mechanical or hydraulic failure. They are finally, tragically brought down by severe, prolonged drought that permanently snaps their fragile liquid robes, by a massive direct lightning strike, by overwhelming pathogenic rot that aggressively breaches the failing Heartwood defenses, or by catastrophic high wind shear that finally shatters their compromised mechanical structure. They are killed by the brutal physics of the world, never by their own internal biology.
Wow, they fight gravity, they fight thirst, and they fight time itself for millennia until physics finally, inevitably wins.
Okay, let's try to synthesize all of this, because what we have uncovered today is just awe-inspiring. A tree is a monument to absolute ruthless biological pragmatism. It minimizes its metabolic burden by relying heavily on dead tissue, literally relying on the empty corpses of its own cells to build its towering structure. It expertly exploits the unforgiving laws of thermodynamics and extreme negative pressure to invisibly move literal rivers of water hundreds of feet into the sky.
It is a profound biological system, unburdened by the senescence of telomeric decay, bound ultimately only by gravity and hydrodynamics. We have seen the active, terrifyingly precise suicidal chemical fortification of the deep heartwood. We've explored the bizarre metabolic shock of the companion cell chloroplasts, acting as pure ATP batteries for zombie pipes, and we've navigated the complex, fiercely competitive realities of root systems and forest ecology that strip away the romanticized myths and reveal the true ingenuity of the organism.
It leaves us with a completely new, incredibly vivid lens to view the world. So here is a provocative final thought for you, the listener, to mull over the next time you step outside and walk down a tree-lined street. Take a moment and look at the trees you walk past every day. Try to vividly imagine the silent, violent, highly engineered hydraulic miracles happening invisibly right next to you at this exact moment. Because when you reach out and press your hand against the rough bark of a mature tree, you have to realize that you aren't just touching a plant. You are touching a dynamically armored, chemically defended shield protecting a pressurized river of water flowing backward against the massive pull of gravity, sustained entirely by a paper-thin, razor-sharp layer of life persisting tenaciously upon a mountain of its own ancestors' bones. Thank you for joining this episode of Audio Notebook.